So this is our CRISPR project. CRISPR, the cure for cancer.
So the first thing is our biological problem. So the causes of cancer are from abnormal alterations to genes. With these changes, cells do not function normally and in turn divide improperly. When cancerous cells go through the cell cycle, the checkpoints used to regulate defective cells are ignored. Those cancerous cells continue into division and create more cancer in the body with the increase of cancerous cells present. An example of a gene that causes cancer is an oncogene. Radiation and exposure to UV are some examples of things that can create defects of cells. Age is another factor for cancer development. Our body is supposed to prevent damaged DNA from becoming cancerous, but older age lowers its ability to do so. Cancer poses a major challenge to humans diagnosed with it. People battling it have a risk of death, along with many other side effects. So, cancer can be a part of almost any part of the body. When cancer cells are formed, they travel through the blood or lymphatic system and can move into any part of the body, but most commonly the bones, liver, and lungs. When circulating through the bloodstream, the cancerous cells can stick to the liver, creating a secondary cancer. This cancer is most commonly metastasis cancer because of the way it spreads. The gene that's involved with the problem is the p53 on chromosome 17. It is a tumor suppressor, as it regulates the cell division. It promotes the repair or apoptosis of cells with DNA damage. So, if it is mutated, it cannot do its job and cancer is created. So, non-genetic treatments, one of them is immunotherapy. It can be very effective because it does not target the cancer, but instead uses the immune system of the patient to try to beat cancer. However, it does not work with every cancer. It tends to be more effective with blood cancers. Immunotherapy is also used once genetic treatments are used and proved to have been ineffective.
The CRISPR-Cas9 system. What is CRISPR and where does it come from? When CRISPR and the Cas9 enzyme work together, researchers can edit DNA sequences and genes. This system was first successfully used in 2012, and it is still the most efficient genome editing system that has been developed. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. In non-scientific terms, CRISPR is a repetitive DNA sequence that was first discovered in bacteria in the 1980s. The CRISPR system that is now used on eukaryotes to edit genes is based on a built-in system that bacteria have always used to protect themselves against viruses. How are bacteria able to use it for their own immune purposes? When a bacteria makes contact with a virus, a piece of the viral DNA is integrated into the bacteria's DNA to create segments called CRISPR arrays. The bacteria now has a record of this infection and can remember the virus to defend itself next time. Once the bacteria detects the DNA of that virus again, it produces two strings of RNA, one of which targets the virus and matches up with its DNA sequence. The RNA in the bacteria promotes the creation of the Cas9 enzyme, which cuts the virus's DNA. This disables the virus and stops the bacteria from harm. What component proteins and nucleic acids are involved in the process? Scientists researched how the RNA and the Cas9 enzyme work in bacteria to develop the CRISPR process of genome editing. Cas9 proteins and a type of RNA called guide RNA are the most fundamental parts of editing genes. Cas9 enzymes have two regions. The recognition lobe binds the guide RNA, but the nuclease lobe is responsible for initiating binding and cutting the DNA. Guide RNA, which is equally as important, also consists of two sections. The CRISPR RNA targets a specific segment of DNA by pairing with it, but the trans-activating CRISPR RNA serves as a place for the Cas9 to bind. How are scientists able to use this system to target specific genes of interest? Bacteria naturally have this guide RNA to target viruses, But in order to edit genes, scientists have to synthetically design their own guide RNA. They do this by merging CRISPR RNA and transactivating CRISPR RNA. This tailor-made guide sequence is designed to match up with a specific piece of the gene that the scientists are trying to change. Its pairs are complementary to the target gene, so it will bind easily. Once it binds, it promotes the Cas9 enzyme to cut the gene sequence. The interesting part is, the cell recognizes that its DNA has been damaged and attempts to repair it. Here, researchers disrupt the natural healing process by adding new pieces of DNA. The old gene has now mutated into a new one.
Okay, so this is problem solved. CRISPR targets the T cells when editing. A human's T cells are immune cells and have the ability to kill cancer. Cancer is a huge threat to one's health. While there is no current cure for cancer, scientists are using CRISPR in the hopes that it will prove to be effective. During the CRISPR process, these T cells are being genetically modified to see or notice cancer better, and then kill it. With an addition of a synthetic gene to the T cell, a receptor protein is added. The receptor protein sees NY-ESO1, or more specifically, a molecule that is attached to cancerous cells. Two genes that can interfere with the NY-ESO1 receptor, and one gene that limits the cancer-killing cells' abilities are removed. Scientists delete the three genes with CRISPR. Then, millions of CRISPR-edited T cells start to grow. These T cells are infused back into the patient, where the now edited T cells will bind to cancerous cells and kill them. The CRISPR-edited cells are being delivered in vitro. The cells are infused into the patient through their genes in almost an IV. The T cells are being edited in labs, not directly in the humans themselves. So how close are we to the cure? The edited T cell genes could make other cells turn cancerous. The cells could then self-destruct. This could lead to the opposite of what scientists wanted. The tissue would then develop cancer, which would spread to the organs. The human would then obviously be affected by this, as they have a deadly disease. This is a future treatment for the disease. Although scientists are getting closer and closer to getting the cure for cancer, it is not a treatment yet. The treatments of cancer include chemotherapy, hormone therapy, hypothermia, immunotherapy, radiation therapy, photodynamic therapy, and stem cell transplant. We are not exactly close to finding the cure for cancer. Some scientists believe we may never find the cure. However, they believe that we are incredibly close to finding an effective method of reducing the incidence in cancer cases. By reducing the incidence, less people will be diagnosed with cancer. Bioethics. So somatic editing only targets body cells, which affecting only the patient being treated. It is also possible to edit germ cells, but that has a larger effect because it changes all the cells in the organism, including the eggs and sperm. This leads to it being passed to future generations.
Since the technology isn't anywhere near perfect, mistakes in one's genes are likely to happen. This could include unwanted mutations. Also, people in the future could want to use the technology for their own personal benefits and their physical appearance.
So our statement is that we believe CRISPR could be revolutionary in killing cancer. However, there is a point where we should draw the line. So we believe that CRISPR is incredibly helpful to edit genes. CRISPR could be revolutionary in curing cancer or even lowering the number of cases. We could save numerous lives around the world. However, there is a point where we should draw the line. In the future, CRISPR will be so much more developed. People could take advantage of this and use it for their own personal wishes, for their children, such as desirable physical traits. And this is our Works Cited page, and then our other Works Cited page. Thank you.